let's talk gene expression. So gene expression, actually, we got two different worksheets on gene expression. One we're going to look at and kind of compare the kind of older method was northern blot analysis to the newer method, which was microarray analysis. And then that actually got supplanted by RNA-seq, which will be a different worksheet. Okay. So first, just I want to kind of refresh our minds about what we're talking about here. And so we're talking about gene expression. Why gene expression is important is because we have the genome's got 23,000 or so genes, 22,000, whatever, something like that. Okay. Protein coding genes. Now those genes we need expressed at certain levels. And some of the genes are expressed in all tissues, some expressed only in certain tissues. Some are expressed only in blood versus brain or versus lungs or at certain stages of development. Or in response to certain environmental conditions are being expressed. Or sometimes they're misregulated in disease, which is pretty much what most of what cancer is going to be about, is understanding how gene expression and what types of genes are changed to give us that state. All right, so let's take a quick look at the worksheet. So this is a hypothetical situation where you're studying lung cancer in this case, and you have a bunch of different patients that you've removed tumors from. While you're in there taking the tumors out, you also biopsied and took a little bit of healthy adjacent tissue. You extract total RNA from the tumors and the healthy tissue to identify any genes that may be misregulated in the tumors. So here we're saying, so the first step is when you're working with your extracted RNA, what can you do to make sure you're only evaluating protein coding genes? Okay, so for this, what you want to think about, and this is an important kind of just conceptually a step, is remember our RNA is, there's lots of different RNAs in there. The ones that are for protein coding genes are the mRNAs. Okay? But remember, ribosomal RNAs, we got 80% of the RNAs in the cell are going to be ribosomal RNAs. And so what we want to do is we want to pull out the mRNAs. And in order to do that, we're going to take advantage of one of the features that mRNAs have that other RNAs don't have. And that's going to be what's called poly-A selection. Okay? And so that's from this right here, where we use oligo-DTs. Overall, this is called poly-A selection. This is using a column-based method. Most of them are some sort of column-based methods. Most of them are going to take advantage of having, you're going to have a little stretch of T's that your poly-A tail can bind to, okay? And then you'll wash away everything else, and all that you're left with are your mRNAs bound to these little beads with these T's on it. And then we can wash off the mRNAs with some other type of solution, like a oops. And then we can enrich for we can enrich for our mRNAs, which are representative of our protein coding genes. And so what this would be what can you do to make sure you're only evaluating protein coding genes? We can poly-A select for our mRNAs. Okay. Now, after isolating the mRNAs, you plan to use a microarray that is spotted with sequences that are complementary to the human protein coding genes. Before applying the RNA samples to the array, you need to label the cancer RNA with red fluorescence and the healthy tissue RNA with green fluorescence. What do you need to do to prepare your RNA samples before applying them to the array? And so this is that next slide is we need to do what's called cDNA synthesis. And so what you need to remember is that there's only a very few places do we actually use RNA as the thing we're manipulating and doing experiments on. Most of the time, we need to convert our RNA into DNA. And when we do that, it's called cDNA or complementary DNA. Okay? To do that, what we need to use is this enzyme called reverse transcriptase. We're not going to worry about the process too much. All that I want you to know about is that what we get out of the end of it is for every RNA we get put into the tube, we get a cDNA out of it. Okay? And in the process, we can label this with fluorescence. We should do that in red. Okay, we can have fluorescently labeled nucleotides to convert our RNA into DNA and also incorporate our fluorescent tag at the time. So what do you need to do to prepare your RNA sample before applying it to the array is we need to convert to cDNA with reverse transcriptase and label with fluorescence while we do so. So while we're converting into cDNA, we'll label it with red or green. Okay, so in this case, we're going to be making the cancer is going to be red and the healthy tissue will be green. Okay. You apply the fluorescently labeled RNAs to the microarray shown below. Okay, now we're going to start asking some questions. So let's take a second to remind ourselves what we're doing when we do this. Okay. So remember, on the microarray, what we're going to have is little stretches of nucleotides that are complementary to, as we're told in this example, the spots on the slide are complementary to the protein coding genes in the human genome. Now we're going to take our RNAs from our cancer cells and from our healthy tissue cells, and we're going to convert them into cDNA, labeling them with red and green. So now, remember that spots on the slide are complementary to the protein coding genes, meaning that A to T, G to C complementarity. And so whatever we have in our RNA pool, if that gene is present on the microarray, we'll have the opposite or the complement of it. We're going to apply both of them in equal quantities to this array. Okay, and then we're going to be competing. And we're going to be getting some spots where we're going to get more red cDNAs binding to it. There'll be some spots we get more green RNAs or cDNAs binding them. And there'll be spots that we get equal amounts, and then there'll be spots that we don't get any. Okay, and so that's what this is trying to, this worksheet is working through. And so remember that when you have equal volumes or equal quantity of red and green fluorescent, how it is visualized comes out as yellow. Okay, so with that, we can look at the first question. And so here's our array with a bunch of different spots. Each one's for a different gene. Some are green, some are yellow, some are red, some have no color. Okay, so beta-actin, it's a structural protein that's equally expressed in all tissues. Okay, so this is a special type of gene. We use these as controls because what we can do is we actually can normalize the reaction to beta-actin because we know that it's expressed in all tissue types. But these types of genes are what we call housekeeping genes, and they're ones that are on at equal levels in any tissue. Okay, so if it's equal in any tissue, okay, what color spot would you expect this to be? Okay, we would expect this to be yellow because equal amounts of red and green fluorescence visualizes as yellow. Okay. So now let's kind of consider this. Okay. So now we're looking at cancer now. So tumor suppressor genes, we will learn in the chapter on cancer, are genes that play roles in regulating cell cycle. Okay. They're often underexpressed in cancers. So often they are responsible for, when we'll talk about this later, is when the cell goes through its cell cycle. Remember G1, S, G2, M, over and over and over again. Before each of those stages, before going from G1 to S, there's what's called a checkpoint. And that checkpoint, the cell stops what it's doing, well, not everything, but doesn't proceed through cell division until it's made sure everything is okay, right? Often, not all tumor suppressor genes, but often tumor suppressor genes regulate that process and kind of slow things down when they need to be slowed down. So if you lose control of your tumor suppressor genes, then you're not having that slowing down process. And so cells proliferate faster than they should, which is kind of the definition of cancer. So if these genes are underexpressed in the cancer, okay, what color spot would they be? All right, so let's think about this again. So if we're putting red for cancer and green for healthy, we put equal amounts on there, but it's underexpressed in the cancer cells, that means that it has less than the normal sample. So if it has less, that means I'm going to get more green cDNAs binding to that spot than I'm going to get red cDNAs binding to that spot. And so if I have more green than red, I'm going to see green. Okay, so with this, one thing to keep in mind is that you don't completely, the gene doesn't have to be totally off. And so while the cancer may still have some expression of that gene, the fact that it is less expression means we're going to get more green fluorescence binding there. So in that way, that's going to make our spot green. Okay. Because there is less expression in the cancer, so more green cDNAs will hybridize, what we say, hybridize to the array than red cDNAs. Okay, now oncogenes, we're going to learn, is another class of genes. And these often drive the cell cycle and contribute to cancer when there are more RNA transcripts than what's found in a normal cell. So in this case, if there are more, in this case it tends to be upregulated or increased expression. And so in that case, compared to normal, I have more red than I have green. So my spot is going to be red because the cancer expresses the gene, which is red, more than the healthy tissue, which is green. So more red cDNAs will hybridize and green, which equals we'll have a red spot. All right, so these are just a couple conceptual questions about this stuff. So what are the advantages of studying gene expression using a microarray compared to a northern blot? So I showed you a northern blot here. Okay. So remember, when a northern blot we're doing is we take RNA from each of these tissue types, and this is one of the times that we're actually using RNA rather than converting it to cDNA. We run the RNA through a gel, and then through this process, that's why it's called a blot, we're going to transfer that RNA to a membrane, and now we can probe that membrane for a particular sequence. Okay? The way we're going to do that is we're going to make a radioactive probe for whatever sequence we're interested in. And in this case, this is taken from cystic fibrosis, looking at the CFTR gene. But what we can do is, so when we do that and we hybridize on this gene onto this membrane, we then can detect where we have that sequence occurring using X-ray film, because the probe is radioactively labeled. Wherever it hybridizes to the RNA, we can then put a X-ray film over and it'll expose it. And so we can see here we got pancreas, lung, and testis expression. Now, this is a really good technique. We can look at gene expression, we can tell relative amounts, we can see that we have more expression in the pancreas than we have over here in the testis by the relative intensity of the bands, but it's not a quantitative thing. And the other disadvantage that we have here is that we can only test this one gene. Okay, if I'm going to go and want to test a different gene, I have to go and get all of these different tissue samples again and do new RNA extractions, make a new membrane and a new probe, and probe it with a different gene. And then if I want to do a different gene, I gotta do it again. With a microarray, we can evaluate all of the genes simultaneously. Okay, so with a microarray, well, with a northern blot, they're limited to only evaluating one gene at a time, while microarrays can simultaneously evaluate thousands of genes. Okay, the last question. Right, microarrays are really, really powerful, but there's some limitations to these. What are these limitations? Okay. The first is that, and I'm going to draw this out a little bit to illustrate. The first is you can only evaluate what you think of to put on the slide, put on the array. Okay. And so if you were to think about, I gave this example in class, say you have a gene, and these are your exons, okay? And you have your array, and you spot in that little spot onto that spot there. Okay? And you got this spot here is that piece of the exon. You got this spot here is that exon. Okay, so if you're looking at expression levels, this gene may be expressed normally, but what may have happened is you may be retaining this intron. Say you have a mutation right there, okay, that keeps you from cutting out that intron. So normally when you would transcribe this gene, you would transcribe it, you'd have your GCAP, your poly A tail, you'd splice out your introns and splice together your exons. But if you got a mutation at the site that you normally splice your introns out with, you're going to keep that intron. And so your disease state that you might be evaluating could be caused because this intron is in the middle of your mRNA when it shouldn't be. And you would never detect that on your microarray because you'd see that that first spot, that's expressed normally, and that second spot, it's expressed normally, and that third spot, it's expressed normally. But you would never actually capture that the intron is in there because the intron is not spotted on your slide. Okay, so in that way you can only test what you think to test, and you don't get any other information. And then the last thing is kind of a technical thing, is that different sequences have what are called different hybridization efficiencies. Efficiencies, which just means the array differently. They stick to the array differently. And so you could get results that aren't actually true. Okay, so you always have to do some experiments to follow up with a microarray experiment to validate the fact that yes, the expression differences you have on your microarray are actually real expression differences. Okay, so this is, and then the last step of doing expression analysis is RNA-seq, which that will be done on a different worksheet.